Alright, today we're going to discuss our first major classification of macromolecules in the human body, and that are proteins. Now, proteins are very important to the human body. They have vast number of functions, and we're going to discuss the different functions of proteins in a minute. Now, this lecture is part one of a two-part series. The first part, we're going to look at the building blocks of proteins, which are amino acids. The amino acids are the monomers that we link together to create these gigantic molecules known as proteins. The second part of this lecture is going to look more or less at the structure, the three-dimensional structure of proteins, and the different capacities that proteins play throughout the body, plus we're going to look at how we analyze proteins in the second part. What you see on this front page here is a computer-generated model of a protein. This is called the ribbon model of it, and you can see all of these little scrolly little ribbons and so forth and turns, and that essentially is the final three-dimensional structure of a particular protein that is computer generated, and we'll talk about these ribbon model structures a little bit later in the second part of our lecture. Okay, so what we're going to do now is we're going to look at basically the function of proteins. IB expects that you are become very familiar with the functionality of proteins and how they work in the body. First of all, proteins can be used for structure, to make cells rigid and three-dimensional, to make them sturdy and create a nice scaffolding within the cells for which it to function correctly. Proteins, as far as structures go, they are also muscle tissue. Muscle fibers are proteins and give our body structure and strength as well. Enzymes are also proteins, and what an enzyme is more or less a catalyst, but it's a biological catalyst, and just like all catalysts, they are used to speed up a chemical reaction by lowering the amount of activation energy required to initiate the reaction. We'll discuss more about enzymes later on, especially the HL class. We'll become very familiar with enzymes, how they work and function, and the kinetics that goes along with them. Hormones. There are different types of hormones, but proteins do make a classification of hormones. Hormones, what they are, is they are just molecules that communicate. They're like messengers. They are delivered throughout the bloodstream, and they communicate between one cell and another cell, or between cells, tissues, or different organ systems that cause these cells or organs to respond and to do certain things. So hormones are very important as communication molecules throughout the body. We also use proteins for protection, and this really implies with the immune system, our defense system of our body. Our immune system is very complex. There's a lot of proteins involved in our immune system, but a great example of a protein in the immune system would be antibodies. Antibodies which search out, find foreign materials, either they're viruses or bacteria or other foreign molecules. They find those, they bind to them, and they cause other cells to come and destroy those foreign materials. So antibodies are very important. We also use proteins in transporting other molecules, either within a cell, or we also use transport proteins in the bloodstream. And we'll be discussing transport proteins in the blood, such as LDLs and HDLs, a little bit later when we get into cholesterol. Alright, so there are some basic functions. To get more information, go ahead and get into Chapter 13 in your IB textbook, and they'll go over this some more with you. Okay, so what we're going to do now is we're going to look basically at our amino acid structure. Now, I'll remind you that the amino acid is the basic building blocks of proteins. All proteins are made up of amino acids, and we link amino acids together to make these gigantic proteins, and we're going to show you how we do so later on in this lecture. Link these amino acids together to begin to make proteins. So to begin with, to understand something that's very important is IB expects you to be able to draw a basic amino acid. And what you see here on your screen is the amino acid that you need to become familiar with to draw. And so we're going to and draw from memory. So what we're going to do is break this amino acid into different parts. It's already kind of done that for us, but we'll look at each component part here. The first we're going to look at is the carboxylic acid group, and that's over here. You should be familiar with those from organic chemistry. Carboxylic acid groups make the amino acid an acid. Okay, so these are acids. Over here on the other side we have the amine group, and the amine group makes the amino acid basic. So amino acids are interesting because they have a basic side to them and an acidic side to them, which is going to create some interesting properties to these amino acids, as well as help us to build proteins. Now, what else is common about all amino acids is they have a hydrogen group here, and this hydrogen group, like the carboxylic acid group in the amino group, is attached to a special carbon right here, and that carbon looks like I made it too dark, but that carbon right there is more or less called the alpha carbon. And the reason why it's the alpha carbon is because we have 4 different groups that are going to be attached to that carbon. The amino group, the hydrogen group, the carboxylic acid group, and then the last one is the R group. Now the R group is a variant group. It means it changes from amino acid to amino acid. It is the R group that makes the amino acid different or unique. There are 20 different R groups that you need to become familiar with. You do not have to memorize all of the R groups, you just need to be familiar with them. Okay? Now, if you have some questions about the basic structure of an amino acid, write those questions down and we can discuss it in class a little bit later. Okay, well, this is an important point before we go on. You can find all of the amino acids in your data booklet. That will be on basically page 17 or table 19 in your IB data packet, which you can use. And it just lists all of the amino acids. It lists, as you'll see here on our next slide here, it will list their names, such as, let's look at the first one here. We have glycine here, and so it lists glycine in your data packet. And in fact, if you have your data packet, you might want to pause this and get that out right now, because we're going to be looking at it a little bit. Again, that's table 19, page 17 in the data packet. And they also give you the 3-letter abbreviation here, and which is nice because we're going to use the 3-letter abbreviation and symbol as we begin to build proteins. Okay, so here we go. We're going to take our 20 amino acids and we're going to further divide them up into different categories. And those categories are based on the R group. And in this video, the R groups are more or less going to be highlighted in this region here. Okay, and each R group creates a unique characteristic about the amino acid. So we have R groups that are considered more or less non-polar, And these are the nonpolar amino acids that you see, because each of the individual R groups here are going to be nonpolar, each one nonpolar. And some of these are a little bit weird, okay, looking, but they are all nonpolar. Now that will have a major effect on protein structure later on, which we'll talk about. Okay. Now, one of the other things that IB is going to expect you to know is to identify all the different functional groups on these amino acids. So again, you have the amino group, you have the carboxylic acid group there on each one of these proteins, and later on you'll see some other groups as well that we will identify. So again, these are the nonpolar amino acids. So they do not have any charges, positive or negative charges, on those R groups. Okay, so now we have another classification of amino acids. We have our polar ones. Now, all of these functional groups, R groups, are all going to be polar. So they are going to have positive and negative charges on them in physiological conditions. And you can see, like serine here, this first one, it has a alcohol group there, which you know is polar. On cysteine, we have a CH group. Erase that a little bit, because so you can see that a little bit better. So you have a CH group, or an SH group, sorry, which is a sulfhydryl group. And then over here, tyrosine, you have an OH group. And it's just that these different groups here on these amino acids are polar. And so they're going to have slightly negative and positive charges on them. And then we have amino acids that are going to be acidic and basic. There are two acidic amino acids, aspartic acid and glutamic acid. What makes them acidic is that they have the carboxylic acid groups there. And the basic ones, they have on their R groups, they have these amine groups here that are going to make them basic on there. And there's three basic ones there. Okay, so again, the amino acids are divided up into three, or really four different groups: nonpolar groups, polar groups, acidic, and basic. And all those groups are based upon their R group. Okay, so if you have questions on that, make sure you make note of that, and then we will look at that in class. Alright, so now what we're going to do is look at what is known as the Zuritter ion. Now, as we mentioned earlier, that the carboxylic acid group here that we'll outline here for you. This is our carboxylic acid group right here. This is acidic, right? And what makes it acidic is this hydrogen right there. That's our acidic hydrogen. On our amine side, we said right here that this was basic. The reason why it's basic is because it has a lone pair of electrons that are sitting there on that nitrogen. Now, what's going to happen in physiological conditions, so in the human body, is that the amine group, which is basic and has those lone pairs, are going to literally attack that H, and in an acid-base reaction. So our base, which is electron rich, is going to attack our acid, which is electron poor. And the result is forming what is known as the Zuritter ion. And the result then is that we have a negative charge on that oxygen and a positive charge sitting on the nitrogen. And that's all there is to it. So a Zuritter ion has a positive charge on the nitrogen, and this hydrogen right here has been added by the attack from the nitrogen to the carboxylic acid group, or the acid on the carboxylic acid, to create this Zuritter ion. Alright, so what we're going to do is I'm going to draw this out. So here's our amino acid. So we're going to start out with our, jump the page here. So we're going to start out basically with our alpha carbon. And our alpha carbon is attached to our hydrogen and our R group. And remember, there are 20 R groups. And then we have our carboxylic acid group right here with our O and our H, and there's our acidic hydrogen, and our amine group, like so, with our lone pairs on the nitrogen. And our lone pairs are going to attack this hydrogen there. Okay, and when it attacks that hydrogen, the amine group is going to be protonated. So we are going to now lose this hydrogen, and our nitrogen now has the hydrogen. We form a positive charge on the nitrogen and a negative charge on the oxygen, and we have our Zuritter ion. Alright? And that's all there is to it. And it is an acid-base attack. Okay, so now we're going to look at a specific property of amino acids. We're going to talk about more or less what is called the isoelectric point. Okay? The isoelectric point of an amino acid is we have a Zuritter ion, and the Zuritter ion has the positive nitrogen and the negative oxygen, which essentially makes the overall charge neutral. So the amino acid is considered to be neutral at the isoelectric point of an amino acid. Alright? Now, each amino acid has a different isoelectric point. In your data packlet, again on page 19, or page 17, table 19, if you want to look at that, you will see in the very right-hand column there is the pH of the isoelectric point. Okay? So what that tells us, for instance, if you look at glycine here, if you look at your data table, you will see that glycine has an isoelectric point of around a pH of 7. Okay? What that means is that at a pH of 7, glycine is essentially going to be neutral. Now, when the pH is going to decrease so that it becomes more acidic, we are going to protonate the OH group with the hydrogen. And so eventually, so below the isoelectric point, at pH is less than 7, then essentially our amino acid is going to be positive charge. And the amino acid itself becomes more acidic. Above the isoelectric point, so as we move to glycine to a pH of 12, then we're going to more or less, the amino acid is becoming more negative charged. And we're going to have a negative charge on the oxygen and no charge on the nitrogen. This is going to happen to all amino acids above the isoelectric point. And above the isoelectric point, therefore, the amino acid becomes more basic. So this is not an easy topic to do. So let me give you some more examples here of this whole thing. So let's use, for instance, let's do alanine. Okay, so alanine basically has an isoelectric point. Let's see here. Draw this. C H R. There's our carboxylic acid. And here's our NH2 group. Okay? Armine. Now, the isoelectric point of alanine, if you look at it, is a pH of 6. Okay? So what that means then, at a pH of 6, this right there, oxygen should be negatively charged, and our hydrogen or nitrogen should have 3 H's, and it should be positively charged at the isoelectric point. So this is what alanine should look like at the isoelectric point, and notice you have the same number of positive and negative charges, so it's neutral. Now, as you move above the isoelectric point, so the pH is going to be greater than 6, then what happens is our amino acid more or less becomes more basic. And so the end result is we have, oops, erase that. This is supposed to be negatively charged. And our amine group here loses a proton. Trying to fit that in there. Loses that proton. There's our R group there. Looks like I'm missing a see, when I'm trying to squeeze it all in here. Okay, let me rewrite that. So carbon, there's your alpha. There's the carbon I'm looking for. My carboxylic acid group is negative, and my amine group there still got the 2 hydrogens, and then there's the R group. So this amino acid is now becoming more basic above the isoelectric point, and more negative. Okay. Now, as we go down the isoelectric point, so the pH is less than 6 for alanine, then essentially we're going to form an amino acid that becomes more acidic. And the nitrogen is positive, and we've added the hydrogen, so this becomes more acidic and more positive. Like so. Alright. Alright, so that's for aluminum. Alright, so now we're going to look at another one. Let's take a look at, let's do aspartic acid. Now, aspartic acid, you're going to have your CH, C, OH. Okay, except we'll draw it in a Zeritter ion, so this is going to be negative. There's our positive. And aspartic acid is an acid group, so acid R group. So it has a carboxylic acid sitting out here. And that's our Zeritter ion. And the pH at the isoelectric point, if you look at your data packet, is 2.8. Now, if it goes above the isoelectric point, then this should become more or less more basic. The solution should become more basic, but so should the amino acid become more basic. So, essentially, it should become more negative. So, the R group is also going to be affected by this. So, our carboxylic acid is going to be still negative. But our amine group is going to lose a hydrogen, so it becomes more negative. And our R group now is going to be deprotonated, and it's going to lose that hydrogen, so it becomes more basic. And so, and negative. So, if we go below the isoelectric point, then our amino acids should become more acidic and more positive then. So we should have our OH group there. Our H, our amine is, let's see here. Plus, and then our CH2, C, and it should become more positive. Our amino acid. Alright, so there's the isoelectric point. This is one of the, I think, the hardest part of the protein section. So if you need some more help with this, we'll do some more examples in class and answer any questions that you have this. You can also read about this in your IB book in chapter 13 as well on the isoelectric point. Alright. Okay, so what we're going to do now is we're going to build a, start building and learning how to build a protein by linking two amino acids together to form what is known as a peptide bond. Now we do so using a condensation reaction. And remember, a condensation reaction is where you take two monomers and you link them together by losing water. And that's what we essentially are going to do with amino acids to make proteins. So we have our amino acid number 1, amino acid number 2. We're going to link them together. Here is where we're going to link them. We're going to remove water from this OH group from the carboxylic acid and one of the hydrogens from the amine group. And those are going to combine together, the OH and the H, to make water. In the end, then, we are going to link the carbon from the carboxylic acid group and the nitrogen here, and we are essentially going to make a peptide bond. Outline this peptide bond for you. Okay, and right there is the bond that is linked together between our nitrogen and our carbon. And that was our carbon there and our nitrogen there. That we have now linked, and this linkage creates our peptide bond through the loss of water. Okay, and now we can create and link multiple amino acids together. This one shows 3 amino acids, and let me outline the peptide bonds here. So we have a peptide bond right here, and we have another peptide bond right here, and we're linking these 3 amino acids together by these 2 peptide bonds. Okay, and again, the result of creating a peptide bond is the loss of water. Okay, now this diagram here shows that very deal, where we're going to again combine our, we're going to take our OH and our H, and we're going to move them and put them together to create H2O. And then that creates this and this forming a bond with each other, which results in our peptide bond that we see there. Again, that is a condensation reaction. We lose water in the process of linking our 2 amino acids together. You're going to have to do this on the test. They're going to give you 2 amino acids, 3 amino acids, and you're going to have to link them together to create the peptide bonds between the amino acids, and showing the loss of water. And we'll practice that a little bit more in class as well. Now, the other part, what this whole thing is showing, erase this here, is that we can break the peptide bond by doing just the opposite reaction, where we bring in water, as indicated by this, and we're going to break that peptide bond, and we'll reform our OH on our carboxylic acid and our H on our amine group, and we have our 2 separate amino acids again. Okay, so through water hydrolysis, we actually break our peptide bond, break the peptide bond using water, and separate our 2 amino acids from each other. Alright, now this next slide shows several amino acids that are bonded together. It shows our peptide bonds as well. Peptide bond, another peptide bond, another one. And then we have our R groups. These are our R groups. There's another R group, R group, R group there. And we've linked all these amino acids together, and we're starting to build a bigger protein. Now, there's some terminology that you need to know here. At the ends of each of our building of our protein, we have a terminal end called the amino terminal, and also the amino terminal is often called the N terminal end, or the N terminus. And at the other end of our molecule, we have the carboxyl terminal end, or known as the C terminal end. Okay, so again we have the amino terminal end, and we have the carboxyl terminal end, the C-terminus and the N-terminus. Now you need to come familiar because you're going to have to label these two ends, the N-terminus and the C-terminus end, okay, on a protein. And along with that, for your information, which we will talk about a lot more in our next tutorial, is that amino acids are always added in biological conditions to the C-terminus. So we always add amino acids to the C-terminus end. That is really important. Make note of that. Again, we always add an amino acid to the C-terminal end, never to the N-terminal, ever. Okay, always to the C-terminal end. All right, so that pretty much wraps up this tutorial. Again, if you need to replay this, do so. Read your textbook, chapter 13, on proteins and the information we talked about here. And we will now end this tutorial, and you can go ahead and start the second part to this tutorial as well. Okay, so we'll see you.